ending the semester with. So this is your central metabolic pathway. So this is typically one of the last sections that you would cover within a biochemistry class, or spend most of the time on it depending on who your teacher was. So it looked to me like we've got people talking about glycolysis, we've got people talking about the electron transport chain, we also have people talking about the energy carriers between all of these, but no one, to the best of my knowledge on what I've read, is really talking anything about the Krebs cycle. Okay. So what I'm going to do today is cover some of the basics, or some of the pieces of the Krebs cycle in my opinion of it, which is ultimately getting away from the strict memorization of, oh, this piece turns into this piece, turns into this piece, turns into this piece, and it uses this, and it uses this, and it uses this. So trying to understand something about how those steps are occurring, and recognize important steps. Okay, so one big question would be, what would be an important step? What might be something that you would say, oh, that's an important step?
Acetyl-CoA transformed into citric.
Why?
Because it's like the most important part.
Within each of these processes, what are we trying to do?
Create energy.
We're trying to get to energy. Ultimately, we're really pushing to ATP. Okay, so, and we'll talk about this as well. So we're trying to generate ATP from ADP. To do that, we have to form a high energy bond. So we have to establish some way to power that high energy bond, or get that energy towards it. To do that, what we're going to end up doing is generating a whole bunch of high energy electrons that can allow us to transport hydrogen ions across a membrane, and then we've got our energy gradient, ultimately a battery, that will then power our ATP synthase, okay, the enzyme that will make our ATP. So within each of these processes, to me, one of the more important steps is going to be making those high energy electrons, okay, trying to get electrons into that higher energy state. So what molecule carries those higher energy electrons? NAD. Okay? So, and then officially, NADH has the high energy electron. NAD plus would be the species that we're going to dump that electron density onto. Okay? So any step that's generating one of those high energy species, be it ATP, because we can make ATP in this process as well, but anything generating NADH, FADH2 officially, there's another one, for some reason it's slipping my head, one of the
GTP?
GTPs are like ATP. It's not a high energy electron. You're just forming a phosphate bond, and I'm going to complain about that too in a second. Q cycle. What is Q? It's not quinoline, is it? Quinone. Anything with a quinone species, you can dump high energy electrons into that. Okay, so when you think about the Q cycle, what they're talking about within the Q cycle is quinone, because quinone can accept those high energy electrons, and we can then transfer them into doing something else. Okay? FADH2 has a quinone type moiety within it, which is what's allowing it to do that high energy electron transfer. Okay? So in all of these processes, what we're really looking for is generating high energy electrons, which means we're looking at redox reactions. Okay? Those are going to be the important steps within it. Okay? Everything else is getting the structure to the point where it can dump out that high energy electron. Okay? Because if we aren't in the proper structural conformation, we can't get rid of that energy. Okay? So to the GTP ATP process. Okay? And not to call people out. I won't call people out. They'll already know once I start circling it. The end of this process is known as oxidative phosphorylation. Drives me up the flippin' wall. Okay? Why? What we're talking about is making ATP. So ADP plus a phosphate goes to ATP. Is this a redox reaction? No. Why do we call it oxidative phosphorylation? It's coupled to something else. To make ATP, do you have to do a redox reaction? Absolutely not. Okay? we've established, or we can take the molecule that generates and catalyzes this reaction, and we can put it into our own false membrane, okay, and put our own false gradient to it, proton gradient to it. No redox reaction is associating at all with this, and we can still generate ATP. Okay, so that's actually kind of a cool process, a whole story for another class. A group out of ASU made a molecule that could absorb light, generate a proton gradient, and power ATP entirely synthetically. Okay? The only thing that's not synthetic is that ATP part. Okay? They took the molecule or the enzyme that makes ATP, the ATP synthase, they embedded that into a membrane that they made, and then they put in all of their extra pieces to generate that proton gradient by absorbing light, and they were able to make ATP. Okay? Using entirely man-made things, except for the ATP synthase part. Right? So it's kind of cool. We could get rid of the entire electron transport chain and just do it synthetically. Okay? The efficiency was horrible, but also kind of beside the point. It's at least proof of concept that we could do it without going through all of these processes. Okay? So when we're looking at these, what you want to kind of recognize, and that's what this diagram is trying to show, is we're shuttling out these high energy electrons into our carriers. Okay? Those carriers will then go through a redox reaction to establish a proton gradient. Once we have the proton gradient, that can then power our ADP to ATP. Okay? So when we say oxidative phosphorylation, what they're saying is this entire process of trying to make ATP requires a redox reaction. Okay? But the formation of the high energy ATP phosphate is not a redox reaction. Okay, kind of makes sense? Just kind of wanted to clarify and address that because I think that's very misleading as far as the terminology goes. And I'm probably going to get on a soapbox and complain about more misleading things throughout this lecture, but I'll try and be nice about that. So what we've got is our metabolic pathway to generate energy. Okay, and we're going to look at a bunch of different steps within this. Like I said, I think a few people are going to be talking about glycolysis. For sure, we've got our electron transport chain at the very end. And then what we'll talk about today is the citric acid cycle and some aspects leading into it. So the first section of the presentation today will be talking about how we get into the citric acid cycle. Then we'll talk about some steps within the citric acid cycle, and then we'll see what happens with time, and I may just be punching through slides, saying here's more information. Okay. I'd be surprised if we get that far into it. Okay. Your citric acid cycle, so some terminology that comes up with this, is the central metabolic pathway. So it's really what's controlling a lot of individual things. We've got a lot of different steps within it, and it produces a lot of individual pieces that can go out into other pathways. So if you remember that slide 4 weeks ago where it just looked like a bunch of lines scattered across the image, and I said when we zoom in we can see individual pieces of it, the entire metabolic picture. If you look really careful at that image, the dead center of it is the citric acid cycle. Right? And that's because pretty much everything within the citric acid cycle can go out to a different metabolic pathway. Okay? So it really does kind of control a lot of different things. Okay? The other big aspect of it is this word amphibolic, meaning
it changes between catabolic and anabolic.
It's involved in both catabolic and anabolic processes. Okay? So we can go both directions, both an oxidative breakdown of nutrients, or we can do a reductive synthesis of biomolecules that we may want later on. Okay? So just kind of an important term to throw out there. This lower right-hand corner kind of goes back to what I was complaining about already. Your electron transport chain, we're dumping in a bunch of NADH. So here they kind of show your citric acid cycle dumping out your NADH, right, which can then dump electrons into the electron transport chain to generate your proton gradient. Okay, it's that proton gradient that runs ATP synthase. It's not a redox reaction in ATP synthase, it's purely that proton gradient. What's happening within the electron transport chain? NADH gives up electrons to the electron transport chain, which means what happened to NADH? It lost electrons, which means it was oxidized. There's our oxidative part of our phosphorylation. Okay? So the oxidative part generates our proton gradient. The proton gradient can then facilitate or provide the energy to get us for our phosphorylation. It's another electron transport chain at the bottom. I think they're showing the same thing, just a different part of it.
Oh, okay.
I'm just going to have to stand and hold that there. Okay, so you can pick that up. Okay, so your textbook had an image in the slides that I thought was, oh man, I'm going to have to keep playing with that projector thing, that I thought was absolutely horrible because you couldn't read anything off of the slide. I'm sure in the textbook it looks a lot cleaner. So what I went through and did is I think I found this on Wikipedia. They have kind of a cleaner-ish image. I'm not quite sure I'd say it's cleaner necessarily because it looks like a giant mess, but it's at least showing each of the pieces. Okay? And at least based on my experiences of talking with other people that took biochemistry from a biologist versus a chemist, what they ended up learning was, well, pyruvate goes to acetyl-CoA using all of these pieces, and then it reacts with oxaloacetate to make citrate, to make cis contase, to make all this, make this, make this, make this, make this. Then it uses all those enzymes, and you have to be able to draw it all out and put all the pieces in. Then you ask them how or why that works. Well, I don't even know that. They didn't ask that question. That's the chemistry part. That's what's driving this reaction. So when we look at metabolism, it's really dependent on organic chemistry and looking at how those structures change. So that's kind of the emphasis that we're going to talk about today, is not so much looking at the names of everything, though I will bring those in, or even necessarily the names of the enzymes, but focusing on each of those individual reactions and saying what changed within the structure. What has to happen and allow for that reaction to occur. So we're going to kind of break it down into those individual sections. Okay? The first place that I'm going to talk about is going to be what I'm calling the on-ramp into our citric acid cycle. It's a bit of a big box there. Really just going to look at pyruvate into the acetyl-CoA. Okay, so that'll be our first discussion, is looking at how we can make the acetyl-CoA, what enzymatic processes are occurring there, how does that mechanistically work. Okay, so this is going to be important because I want to see something about mechanisms. Within the citric acid cycle, there are a bunch of different steps that we could look at the mechanism for. Some of them would be a lot easier than others. The ones that I've decided to pick are looking at the ones with the energy transfer because, like I said already, the whole point of these things is to generate those high energy electrons. So I want to see those, okay, personally. Within each of your own topics, if you find some other process that doesn't involve redox, that's fine. Okay? Not everybody's going to be looking at energy forms. Okay? And you can take a look at those and you can try and postulate a mechanism that would explain what you've read about and observed. Okay? Does it have to be 100% correct? Absolutely not. What I want to get you doing is looking at how the electrons move, thinking back to organic chemistry, and how to apply those terms back into what we're working with here. Okay? So if we pull it out and look at that step, this is the part that I get particularly irritated with Biochem. Okay? We look at our reactants. We've got pyruvate. We're going to bring in NAD+. We're going to bring in coenzyme A. The big part of coenzyme A that's going to be really, really important, which is why it thankfully does get kind of highlighted extra, is that thiol at the end, and in particular the sulfur of the thiol. What's special about that sulfur? Kind of, sort of. It's a good leaving group once it's attached, but before it gets attached, it is a lot like oxygen, in which case it has 2 lone pairs. Try it again. It's a good nucleophile. Okay. We've got those lone pairs. Those lone pairs can be donated into a structure to form the new bond that we're looking at with our acetyl-CoA. Okay. As another result of this reaction, we're forming NADH. So we can look at the structure of our NADH, and we're also picking CO2 out of this reaction. Okay. So the first thing that I would do is look for an obvious piece. CO2 looks like a pretty reasonable structure to start with. for me because I got 1 carbon and 2 oxygens, which means I would go back to my starting material and say, where do I have a carbon with 2 oxygens bound to it? Okay, it doesn't have to have 2 double bonds. I just need to find some carbon that has 2 oxygens because that's going to be the most likely source for the CO2. So where do we see that? Up in the pyruvate, and we'd be seeing that probably right here. Okay, so there's our CO2. So let's think mechanistically, how could I generate that CO2? And I know I didn't draw out very good arrows or charges on these. What's the charge on that bottom oxygen? It's officially negatively charged. So to generate CO2, what do I need? I need to push the electrons this direction. Okay. Which means our central carbon has too many electrons, so what has to happen? Okay. These electrons have to come out. Okay. And this is where, in my opinion, it gets dicey. Okay. Why might it get a little bit interesting? I can't put them there because oxygen already has too many electrons. So I can't shuttle them out that way. I can't shuttle them out there because we've got a CH3 that already has too many electrons. So really the only place I've got to dump it is right onto that carbon. Okay. That's a really, really odd structure. Okay. I'm looking at putting a negative charge on a carbon with no resonance stabilization. Okay. And if you think back to organic chemistry, you were really limited in the amount of carbon nucleophiles, or negatively charged carbons. And that's because they're incredibly unstable, because carbon is neither large to stabilize the electron density or electronegative, I guess nor electronegative. So it can't stabilize that electron density. So right away I say, there's a problem here. I need some place to dump those electrons. Okay. Well, we could try and say it dumps into NAD+. Okay, so we could take those electrons and throw them into NAD+, except what does it become? NADH. It's not dumping in electrons, it's dumping in hydride, H-. That's just electrons.
Does the hydride come from the thiol?
So we could look at the thiol. I like that option too. Trying to find anywhere where we could pick up a hydrogen. Would I expect hydrogen to take electrons from sulfur? No. That sulfur-hydrogen bond is actually more acidic than water. Meaning, our electron density is going to go away from the hydrogen, and we end up with a positively charged hydrogen. This is why I get irritated. I look at this and say, the pieces don't add up. Okay? And that's because they don't. What does biochemistry do? The protein does it. What part of the protein? What contribution is the protein actually doing into this? And if it's not just the protein, it's, oh, there's a cofactor that's also involved in it. Well, what's the cofactor? Okay, well, let's go back to our initial drawing here. What's the cofactor? What piece is missing? Do they tell you? No. Okay, that I find incredibly misleading. Okay, and the reason why is because as biologists, we can come in and just say, I don't care. Biology is going to do it for me. I'm just going to look at the overall pieces. Okay? Really, really irritating and frustrating to me. Okay? So what is actually helping out in this case? Okay, well, if we look at a slightly more developed version of this, we start with our pyruvate, okay, and this is only slightly more developed, and we couple that with thiamine. Was thiamine in our last reaction? No, why not? Because it catalyzes the reaction, it gets reformed at the end. So they say, well, it shows up at the beginning and the end, I don't need to draw it. Not really. You should actually kind of specify things that actually change how the reaction works. That's kind of an important piece. Okay. Further, do they show how the thiamine is actually interacting? Pyruvate turns into CO2 and thiamine does some weird stuff. Okay? Makes some new structure that can then react with the next thing and then reacts with the next thing. There's zero understanding of how those atoms are actually exchanging places. What part of thiamine is actually making those bonds? Who knows? Okay, it's not. Apparently this is a biology room. I'm talking too much smack. Should they staple it to the floor? Okay. So it depends what we're trying to get out of it and what we want to see come through this. Okay? In a biology class, they don't care necessarily about what's happening within the bonds or how everything's exchanging. All we care about is, does it work? What does it need to be there? Let's just say it's there and move on. Okay? Which is cool for biologists, but for someone like me that wants to understand how and why it's working, I want to see all of those pieces. And if they don't show it, it makes it very frustrating because you have to go and search somewhere to say, okay, you're not telling me something. What is that thing you're not telling me? Okay? So what we're going to do is take a closer look at this because it's not even just thiamine that's missing. Okay? What is this giant thing here? Oh, this is awesome. Protein. Is it just protein that's doing something? No, it's a piece that's attached to the protein. That piece is critical to this reaction working. Again, not listed. Okay? It's not just the protein, it's that particular functionality on it that's allowing these reactions. Take it a step further. What is this? FAD. It's an electron shuttler. Why is that not in our previous reaction? Because it's bound to within the protein and it's not allowed to actually move very easily. Okay? It's large enough that it gets pinned. The NADH or NAD plus in our NADH can actually shift and move through our protein much more quickly. So what ends up happening is our pieces come in, the protein wraps around it and lets certain things out of it. Okay? The certain things it lets out are our NADH, our CO2, and where did it go? Our Acetyl-CoA. Okay? So how does this reaction work? Okay? It's all really going to come back to that thiamine molecule, and it's all going to be based on a mechanism. And I know you all love organic chemistry, so we're going to take a look at how this is going to process out. Okay? I will warn you that this mechanism is kind of big, and I'm sure some steps were skipped within it, and I didn't actually draw it out. I stole it from the internet. Okay. But there are some parts that we wanted to address, and more importantly, around the thiamine molecule, because it's really, really important. So what I actually want to do is step back to a particular reaction from organic chemistry. How many of you have seen this? One person saw this reaction. It's fairly standard. What does the T-butoxide do? T-butoxide is actually a pretty horrible nucleophile, and the reason being, so you got a reasonable idea with the negative charge. Negative charge, we could think a nucleophile, but it's a tertiary butoxide. It's now sterically hindered. So to act as a nucleophile, it must make it into our reactive site. Because it's so sterically hindered, we limit the ability for it to navigate into the reactive site, and it's going to react instead on the surface, which means it'll react with hydrogens. Okay? It'll act as a base instead of a nucleophile. What was that, Victor?
Is that going to add an OH?
Nope. So our base will come through and remove that hydrogen. Okay, which means the electrons on that hydrogen-carbon bond go into the carbon, and we end up with not a cation, but the anion. Okay, so the carbanion. It's a good thing I drew it so they all overlapped in each other. The other part that's interesting within this is that each of these carbon-bromine bonds is actually very, very polar. Okay? And given the proper circumstances, what will end up happening is one of those bromines says, I'm tired of sharing with all that electron density there. There's so much there that one of those bromines says, F this, takes its electrons with it. Okay? So it leaves. So let's say it was that top bromine. Now what we end up with is this structure. What's interesting about this structure? What's the charge? Try again.
No charge.
Try again. There is no charge. It is neutral. Okay. Huh? Not, sorry, aromatic?
Radical.
Not radical either.
Yeah, that's two.
Okay. We have 2 electrons in an orbital, and when that bromine left, it left an empty p orbital. So what we've got on this structure, if we turn it from the side, we've got our bromines, we have our electrons, which it's horrible to show it in a bond, but we're going to say it's in a bond, and then we have this p orbital. So we've got now a single atom with both a p orbital and a lone pair. So it has the characteristics of both a carbocation and the characteristics of a carbanion. So what can happen? You line that up with a double bond. Whoops, didn't mean to do that. Take our electrons, attack our pi bond. The pi bond comes out, attacks the p orbital, and the end result you get a cyclopropane. Okay, whoops, and I left off the bromines, sorry. So what we're looking at with this reagent is what's known as a carbene. We have that both negative and positive characteristics within it. So it's actually kind of a neat molecule. Very, very reactive, okay, as far as those intermediates go, and we need very particular conditions to allow for the formation of the carbene. Okay, because our bromine, or that structure initially, we have to form the carbanion. So to form the carbanion, the hydrogen must be acidic. How can we get the hydrogen acidic? We're going to have to make sure carbon is taking the electrons away from hydrogen. The difference in electronegativity is virtually identical, but if we drop on a whole bunch of electronegative elements to the carbon, we get a very strong inductive effect, and we can pull the electron density away from the hydrogen. Okay? Because bromine is now also a good leaving group, one of those can get kicked off, and I can generate my very reactive carbene. Okay? And I can do some really cool stuff with it as far as making my cyclopropanes. Okay? As neat as that structure is, it's synthetically very difficult to make, very high energy state, okay? Not super likely to form as we would predict in nature because of this high energy state. Okay? Turns out we can see it in nature. Where do we see this molecule show up in nature? Thiamine. Thiamine will actually set you up with a carbene on that carbon. Okay? And there's some issues with this overall structure. I didn't actually address it too much or look at it too much. The only part I'm really concerned about is in here. I circled way too many atoms for that, but there's our primary issue. Our nitrogen needs to be positively charged. Okay. There is a decent amount of resonance within this, but that's not going to be our most important aspect. That hydrogen attached to our carbon is very labile, okay? Or as they're saying, dissociable, meaning it's acidic. Okay? I can take the electrons in that bond and I can put them onto the carbon, generating the carbanion. Definitely didn't give myself a lot of space here. So if we redraw just that overall ring part, right, close enough? Well, does the nitrogen want to be positive? Nope. So the nitrogen can take the electrons back, and what have we formed? We have the carbene. Thiamine is an example of a naturally formed carbene, right, that is actually relatively stable. It's that central part of the molecule that is really involved in the electron transfer of that pyruvate mechanism. Okay? That's probably the central part of it, the one part that we absolutely have to have. Everything else, I can probably fudge a mechanism that gets us close enough. Okay? But we have to have this ability in here. Okay? What's special about that carbon? Nucleophile. With that nucleophile, I can attack a positively charged carbon, okay, within the carbonyls of the pyruvate, which can then go through and help facilitate the overall following next steps. Okay, so thiamine's a pretty cool molecule in that respect. Okay, and then of course they go through and abbreviate TPP because you didn't have enough letters dancing through your brain. So, overall mechanism, I was going to try and stepwise it, but I got lazy. What we're looking at in the upper left, we've got our pyruvate, and then we have our thiamine. Okay. What is E1 standing for? True, E1 would typically stand for elimination. What's it standing for in this case? It's our enzyme. Okay, so our squiggly line is just saying the rest of the molecule continues out to enzyme 1. Okay, why do we not draw out the entire enzyme? Probably because it's really big, and then also more importantly, is it actually contributing to the overall mechanism? Probably not. Okay. Depending on what you find online, you might see images where they show amino acids leaning in and trying to stabilize different intermediates. Those are kind of cool images. Not in this one. It starts to get kind of crowded, so you have to be careful on deciding when to show those. So what will end up happening, I remember we said this was our CO2. We need to be able to facilitate the electrons in this bond, now our purple bond, going somewhere. Okay, so there has to be something to help push that. Okay, so the first thing that's going to happen is our thiamine, with what is effectively our carbene, can attack our carbonyl. And you'll notice it's not a perfect mechanism because what happened? Where's that oxygen end up? Turned into OH. How did it turn into OH? It picked up a hydrogen from probably the protein, actually. Okay.
Why would it
So-
be the one that left off the carbon?
Where it left off the pyruvate?
No, the hydrogen that came off the carbene.
It's possible that that's where it came from. That's probably why they didn't bother to show where that hydrogen specifically came from. Your protonation, deprotonations happen so fast, and you saw this within sapling. We typically don't even, we show them in organic chemistry. In biochemistry, we tend to say, oh, it just happens. I'm not even going to bother to show the arrows. I'm just going to get rid of the hydrogen or put it on magically. Okay. So what's happening is as it attacks, our oxygen comes out and picks up a hydrogen, potentially from the protein, okay, or from potentially water in the solution. Why would we want that to happen? If it stays negative, it could potentially go backwards and kick our thiamine back out. We don't want that happening. Okay? So we want to kind of stabilize that intermediate by putting on that proton. Okay? Now that our thiamine is attached, okay, here's where we get our cool step. We can take the electrons, okay, off of our CO2, we can dump those in to make our CO2, and now we've got some place that we can shuttle those electrons. Because when I go through to make that double bond, sure, this carbon has too many bonds, but I now have a weak polar bond to break within the thiamine. Okay? So the thiamine is now facilitating a temporary place to dump those electrons so that I can then delay and wait for those to be picked up by something else. Okay? Remember how I said we usually don't care about the protonation, deprotonation steps? If we take a look at the next sequence of steps, they're all protonation, deprotonations, and not only are they protonation, deprotonations, what's the difference between this guy and this guy? The one on the bottom right.
The one on the bottom is being attacked by a single bond.
Our nitrogen dumps electrons in both cases to the exact same space. The pi bond breaks and reacts with something. The only thing that's different is it's something else. Okay? In this case, it's instead of it being an acid, it's reacting with the sulfur. Okay? The sulfur of our lipoic acid, okay, which is bound to a secondary enzyme. So why do we show that intermediate stage? It's quite possible that what's happening is it's time-wise that has to form before the second enzyme can actually get into place to allow for the reaction to occur.
Is it because it's so hindered?
You got to be careful with hindrance because you're now looking buried inside a molecule. So
Because it just looks-
everything is going to be hindered. And that's one of the nice things with proteins is you can then force things near each other regardless of the hindrance. Okay.
So the nitrogen is just basically.
It's oscillating back and forth. Okay. That nitrogen, and I definitely meant to look this up, but I'm pretty sure that structure has a very interesting name associated with it. I think it's a Schiff base.
How did we get an extra methyl group on there?
Extra methyl group where?
From step 3 to step 4
Where's the extra methyl group?
on the top.
Oh yeah, on the top.
Next to the R.
Next to the iron.
Here?
Up. There you
Right,
go.
left hand corner.
Oh.
There's no methyl group there.
Yeah. That's a typo on their part, and officially my part because I didn't catch it and put it in there. Okay. It's not that they got an extra typo, they actually got rid of it. They forgot to put it on there. They forgot to put it on there? Did I say that right?
Yeah.
Okay.
They forgot to include.
So our thiamine is facilitating that electron transfer. We're dumping the electrons out to this sulfur group. Okay. What we've got there is a disulfide bond. Okay. Why would the disulfide bond react in this way? Sulfur is very, very large. It has all sorts of crazy chemistry. It can act as both as an electrophile and a nucleophile pretty much all at the same time due to that size. So one of our sulfurs acts as the electrophile, and the other sulfur will pick up a hydrogen to stabilize its negative charge. Okay? You could equate this to the bromine reaction with alkenes. Okay? Where if we look at how bromine reacts with an alkene, what happens? The alkene attacks. Okay. And then we can get it coming back the other direction. So we get something kind of similar in this case. Which button do I want to push? Let's try that one. Okay. Once we now have this massive complex, we can have the electrons on our oxygen come back down to reform the pi bond. Okay. That allows us to free up and kick out the thiamine. And we end up with our thiamine, not shown, but we'd end up with our thiamine right back to where we started with it. Okay. So what it's acting as is a temporary electron dump, facilitating the electron density shifts around that reactive site, and then it goes right back to where it started. So it is strictly a catalyst in this case. Okay. What we're then left with is our acetyl group with a sulfur attached to it. Why is a sulfur a really cool thing to have attached to an acetyl group?
Because it's a good leaving group.
It's an awesome leaving group. So our coenzyme with its sulfur, which has probably been deprotonated again by the protein, can now come in and attack our carbonyl. We'll go through our tetrahedral intermediate state because our pi bond's the weakest bond. When it comes back down, it'll reform and kick out. And this is where I would kind of change my opinion on how that reaction occurs. When it comes back out, those electrons can pick up a hydrogen, again from our protein. Okay, once we have that molecule, that gets shuttled around to actually react with the NAD8 or NAD+. Okay, so that's the other big part of this. In the course of this whole reaction, we haven't even shown the NAD+ converting into NADH. Okay? So, questions within the mechanism?
Would it really get the hydrogen from the acid since you already took hydrogen like several steps before when you get it from?
You're going to get it from
make almost like another
some
catalyst?
amino acid
But wouldn't
within
you
the
get it
protein
from-
structure.
Wouldn't you get it from the sulfur? So that way you remake the catalyst?
So, remake which catalyst? This catalyst? Where are you talking?
With enzyme 2 attached to it.
Enzyme 2 attached.
Enzyme 2.
Yeah.
Enzyme 2 is attached to what?
This group, yeah.
So wouldn't it get it from there so that way you could make that product to...
So, if you try to get it from there, this sulfur becomes negative, okay, and this sulfur has a hydrogen on it. We'd end up with the exact same state.
It won't react.
It's
So
not
then-
going to do anything
so then
different.
After you do this part, what's really gonna happen is
Once we've got our
the
lipoic
conjugated
acid,
acid's gonna take that H off of the S and then make it?
conjugated acid.
Well, once you remove that hydrogen.
So yeah, so where we'll be, I know that's going to irritate you, but tough.
I'm not irritated.
Once we're at that species, the lipoic acid can shuttle back around to our NAD+.
Oh, and grab the hydrogen. Or,
Make sure I do this right. Positive.
there you
Okay. Our sulfur can take electrons from one of those hydrogens, so we kick off an H+. But now what happens is that sulfur has too many electrons. So what will end up happening is it'll take those electrons with the hydrogen, we'll attack that position of our NAD+, and we'll form our NADH. You see it?
Yeah.
Okay. As we'll probably talk about in a little while, whether it happens here or somewhere else, I probably wouldn't have thought of using the lipoic acid. I probably would have shuttled the hydrogen in a lot earlier in the mechanism instead of actually having it go through the lipoic acid structure. Okay. I'd be wrong. Okay, so the lipoic acid is actually necessary to allow for this conversion to occur. So we end up forming all of these kind of reactive intermediates to then shuttle through over ultimately to our sulfurs. Okay, so just like cysteine was really important for other functionalities, it's going to be important for our disulfide bonds because those sulfur groups can allow for shuttling of electrons. Okay.
So NAD plus converts to NADH at the very end?
Converts to NADH at the very end.
Yeah.
Yep. And you're going to love this too. That's saying it goes to NADH. Effectively, the lipoic acid goes to actually FAD to make FADH2, which then dumps it to NADH.
So there's another
Right.
step.
So there's actually another step within that. Okay.
But all
Miracles,
this is, like,
yes.
Happening
And all of this is happening ridiculously fast.
instantaneously, pretty much, right?
Yeah. So, I mean, it's absolutely fascinating that biology can crunch through and do this. And to me, the part that I think is kind of frustrating is that if it's so interesting that this is happening, why is it never discussed? Okay? Why, when we talk about it, we just say, oh, it just happens. Okay? We can explain how it happens. Let's go through that explanation and actually see what's going on
For
with it.
biologists who don't understand organic chemistry.
Well, but see, that's the thing. All of them need organic chemistry.
That's true.
So, yeah, I guess it's a story for another day. The last thing would be once we've got the acetyl-CoA, and I'm calling this the on-ramp into the citric acid cycle, the acetyl-CoA can react with oxaloacetate through yet another enzyme, or citrate synthase, to generate citrate. Okay. That reaction, what do you think? Big and complex or straightforward?
Big trouble.
Let's take a look at what changed. Okay, what I would want to identify is, first off, do we see coenzyme in the final citrate structure? No, it's actually over here. So somehow or another we have to kick that section out. Okay, so what I want to look for is where is this piece in the product? This I also find kind of irritating. They did some rotating on this.
To the carbonyl.
To the top of the bottom.
Oh yeah, officially it could be the bottom too. Okay, I would put it at the top because that's trying to keep that linear thought process. Okay, so what we've got circled in blue are the pieces that swapped out. Okay, so what attached to the oxaloacetate? Where's the carbon that was reactive within our oxaloacetate? So here it's our purple carbon. Where's that purple carbon in our oxaloacetate? It's our carbonyl carbon. How's the carbonyl carbon going to react? Positive or negative? Should be positive because
Electron withdrawing from the oxygen.
you've got electron withdrawing groups from the carbonyl. What atom attached to that carbon?
Another carbon.
Another carbon. Which carbon? RCH2. Is that carbon nucleophilic?
No,
What has to happen?
you have to make it.
It needs to be a carbanion. You're absolutely right. It needs to become a carbanion. So it needs to take electrons from hydrogen. Okay? So let's erase one of those hydrogens. We'd end up with this structure. What did we say about negatively charged carbons earlier? It's pretty horrible. Except
If you have an enzyme. Oh,
if we have resonance stabilization. What is that resonance stabilization?
C double bond or C double bond C.
Most cold.
Mm-hmm. Are you getting the right idea? If we added that hydrogen there, does that help? What is this functional group? An alk what? What is this functional group? It's an alcohol. It's the keto-enol tautomerization. I know that was everybody's favorite section in organic chemistry. Okay. The keto-enol tautomerization is actually what's facilitating that carbon becoming nucleophilic. Okay. If we did not have that resonance stabilization, our protein's going to have to throw in some wicked reactive species to be able to facilitate getting that carbon to be negative. Once it's negative, it can now go through, let me go through and erase some of these arrows. Once we have that negatively charged carbon, it can now go in and attack there. What would still be attached to it? Sorry, let me clarify. What would be attached to the carbonyl carbon of our pyruvate?
The oxygen.
Our carbonyl oxygen, yep. That CH2, yep.
Oh,
Our coenzyme sulfur structure. Is that in our answer?
no.
No. What happened?
It can kick off as a catalyst.
It was kicked off and replaced with
The CO.
OH. We're reacting it with water. Water is a better or a really good nucleophile. We can have water attack that carbonyl and kick out the sulfur, do our proton transfers, and we now have our final structure. Is this a redox reaction?
No.
No redox. Okay, there was no strict electron transfers between atoms. What we looked at were electron motions, but never a strict transfer of electrons.
So sulfur would be connected up at the very top and then kicked off by the water?
Yep. So what we had addressed already was getting us to this stage. Now water can come in and kick out our sulfur. Okay. No redox. So the takeaway is understanding how these reactions work. Okay. That is critical as far as I'm concerned. Otherwise, all you're doing is memorizing. You don't need me. Okay. The reason behind this is trying to look at and understand how and why these things function the way they do, because then you can manipulate and change the conditions, study better things, come up with ways to prevent the reaction from working. Okay? That's what biochemistry is trying to do. You're trying ultimately, in a lot of cases, to generate drugs. Okay? Why do you want drugs?
Drugs are good.
Okay? You get a couple different reasons we can run with drugs. Drugs so that you can mess with your brain so that you go all crazy. Okay? Or we could actually do drugs that mess with your brain so that you don't go all crazy. And then we could also do drugs that will actually go through and kill things, like cancer cells or other bad cells, bacteria within your body. We need to come up and understand how those enzymes work so that we can target those things and kill them. Okay? If it's consistent across all patterns of life and everything does the exact same thing at the exact same time, we want to make sure that our drug does not target that. Because otherwise, you die. That kind of sucks. Okay? Yeah.
So energetically, when we're looking at the delta of G,
is that a high energy or low energy compared to the one that we just did before? With diamine?
What do you mean?
Like energetics wise.
Energetics wise, this is a release of energy.
But is that a high? Is that a high?
Big energy difference versus the other one? I don't know that one.
Okay.
Yeah.
Because it seems like the other one just took.
It has numbers,
Okay.
and when it has numbers, I tend to kind of get blurry in the vision. Your textbook definitely summarizes and says, in this step, it's this much energy, this much energy, this much energy. Both endothermic and exothermic for multiple steps, for different steps. In any other class, yeah, that might matter. I don't care about it. Okay. One of the other big things that we can get out of this reaction is catalyzed by your citrate synthase. So what this enzyme is doing is bringing in the pyruvate, your acetyl-CoA with the oxaloacetate, and that enzyme is what's facilitating that bond formation. Big important thing is that is an allosteric enzyme, and it responds to NADH and ATP. Why might it respond to NADH and ATP?
Because one's providing energy and the other one's taking it.
Yeah.
What does NADH end up going to ultimately? To do what? Sorry, we're going to skip back a lot of slides. Go all the way back. All the way back. What do we do with our NADH? We use NADH to transport electrons to make ATP. Okay. So NADH is effectively ATP. Both of them are an energy source.
But why do you need two?
Oh, because
They're going to do different things. So the phosphorylation is one energy source. The NADH is a redox reaction.
Does it have to do with the amount of energy that's given, or?
There's an interesting idea. What if you already have tons of energy? Let's say you have tons of NADH and ATP floating through your system. Do you need more? Should you be running the citric acid cycle to produce more? No. So what's going to happen? You're going to take that NADH and ATP and have that dump back into your cycle to turn it off. Okay? Your citrate synthase is inhibited by NADH and ATP. If we have a bunch of extra energy floating around, we want that energy to come back and do something useful. If it's not doing anything useful, and it better stop being produced. Because ultimately, if we give it too much energy, now we've got a bunch of energy that can go out and do other things that we didn't want to happen. Think about organic chemistry. When we run reactions, heat is a pretty good thing to add to our reactions because it speeds the rate of the reaction. Does it speed the rate of just your reaction?
Yeah.
It speeds the rate of all reactions. So if we dump out tons and tons of energy, right, that energy can then go out and do all sorts of other things that we may not want at that exact instant. Taken conversely, what happens if you all of a sudden need energy? Well, if you've already dumped it all out there and it's busy doing something else, you now don't have the energy for that process. Okay? Running away from some predatory animal. Okay? So within these cycles, we actually have shutoffs that prevent the production of more energy.
So excess of NADH and ATP shuts off that.
Will shut off that particular enzyme. Okay, so that was our citrate synthase. So some exam questions that we come out of this. Wow, that was supposed to be one of two presentations.
And you're almost at it.
Yeah, I know. Thank you.
This is organic.
Okay, yeah, it's organic, so I get more excited about it. The other part of it has more organic in it too. All right. So role one, or question one, what role does thiamine play in many redox reactions?
You and your tricky question. I don't know.
What charge did our thiamine pick up with? Negative. For it to be negative, it's either acting as a base or as a nucleophile. Both of those are options. So now the question is, which did it act as? What did our negatively charged thiamine molecule attack? a carbon, it's a nucleophile. If it attacks a hydrogen, it's a base. It attacked the carbon. So what we're looking at is it acting as a nucleophile.
What's a Bronsted base again?
Bronsted base is an electron donor to a hydrogen ion. And it's officially a hydrogen ion, H+. Question 2, thiamine is an example of a naturally stable carbene. Hey, good. Sometimes there's some sort of softball questions. Does the conversion from ADP to ATP require oxidation? No. So I did throw in a bunch of other things that could possibly show up. Okay. Yeah, NADH does help for that conversion, but it's not necessary. We're looking at, what is the name of Q? Oh, it's ubiquinone? Okay, ubiquinone, okay, can be used, again, to generate our proton gradient, but it's not strictly necessary. Same deal with our ATP synthase. ATP synthase is absolutely necessary to make ATP, but does it involve redox? No. All right. Question 4, which of the following does not contain enolizable hydrogens? Yes, organic chemistry. So what we were looking at for our enolizable hydrogen, we go back to our acetyl-CoA. If we were going to enolize this structure and turn it into its enolate,
One of them
would be that guy. What changed? What did we need to have in our initial drawing there?
arrows.
Take away hydrogen from the carbon.
Take away a hydrogen from the carbon next door. So what do you need to look for within your structure? Carbonyl, and it has to have a carbon next to it with hydrogens.
It's the
Or it's the first one.
So that one for sure doesn't have a carbonyl, so we can't call that one enolizable.
So that's the
What's the deal with the bottom right? We don't have that carbon with our hydrogen. That's also not enolizable. So it's C and D. Or there's no letters to it because I real quickly made this question this morning.
So, answer C and D?
Yes.
Because you need a...
I had issues formatting questions. Give me a break. So trying to put that together, I didn't put through all the A, B, C, and D. What I would probably do in this case is say, these are your choices, 1, 2, 3, and 4. 1 does not contain enolizable. 2 does not. 3 and 4 do not. 3 does not.
It's a terrible question.
So you can get all of those kind of...
So you need a carbonyl with a methyl group?
You need a carbonyl, and it doesn't have to be a methyl group. It needs to be a carbon with a hydrogen. So the critical parts of our enolizable situation is what's now highlighted in purple. That's what's required.
So you're not going to ask like just D or just C. You're going to ask, say.
That's all. I still have to format the multiple choice answers off of that, but that would be the question with those structures. I will not change the structure.
I do like a short answer for all of them.
I could do it as a short answer too.
So the hydrogens on the ones that you circled are not...
There's
no
no
count?
carbon.
There's no carbon, so look back to the bottom section, which I'm now highlighting in blue. The pieces that we need are those, what is that, 4 atoms? We need the hydrogen bound to a carbon, bound to the carbon of a carbonyl with the oxygen. We have to have all 4 of those pieces. If we're missing any of those 4, you cannot enolize. And if you go back and look at those 2 structures, you're missing the one on the left, you're missing the carbonyl. The one on the right, you don't have that carbon with its hydrogens.
Got it.
I was a bit tricky on that because I threw in those hydrogens. Yeah, so watch out for that.
You showed the hydrogens.
Yeah.
Watch it.
Last question, why might it be important for citrate synthetase to be allosterically affected by NADH?
Inhibition.
Okay, so one question is it inhibition or activation? So inhibition, why?
Shuts it off. You don't
So we can not continue producing that excess energy if we aren't going to use it. Okay, so it just has to do with that process. That was like an organic lecture, really. I mean, I only got through like 10 slides.
It's okay.
So those are your exam questions coming from that. Do you guys have any questions? How many of you have not signed up for your presentation time?
I gotta do that.
You should probably do that soon.
Yeah.
Okay. That sign up is on the discussion boards. Okay? You'll have to go through and check the time or the dates. It's 2 presentations per day. Okay? If there is a missing presentation, then I will look through it and I will show you one. Haha.
There's one on Monday.
Yeah. So what you'll probably end up seeing is more on the Krebs cycle, because I think it's actually kind of cool to look at what changes there are in it and to change your perspective on how you're looking at it, as opposed to just this step, this step, this step. What's happening in those steps? What can you identify as changing? Really bringing in the chemistry aspect into biochemistry through the Krebs cycle, as opposed to just, here it does it. Okay? There's no way in heck I'm going to be able to do the next sequence of slides in 10 minutes.
You can show us how
So-
far you went through your really short presentation.
It's up 7 up.
What are the differences? So what I'll probably end up doing is restarting this presentation at a later date, because there's guaranteed to be open spots. So we'll re-go through this, but we can get kind of the idea on what's happening. What's the one step we've talked about so far?
Pyruvate to acetyl-CoA with oxaloacetate.
We really did this step, and we also kind of looked at that next step. Okay.
All after that.
Yeah.
I was retarded.
I think it was awesome, personally. Because that stuff is what's actually happening in an individual cell at pretty much all times. I mean, that's pretty impressive. No. But I'll take points away if you don't. I'm kidding. We can take pyruvate and we can react pyruvate. Whoops. I did the exact same thing, biochemist. Damn it. We can take pyruvate and react it with bicarbonate, and we can make oxaloacetate. Okay, so that's what that other arrow is showing. So if for some reason we're probably has to do, actually it's a good question, I should look that up. See why we would need to do that. Is it gluconeogenesis on why you need to do that? Oh, because you have an excess of pyruvate, right?
Yeah.
Cool.
And then when there's too much oxaloacetate in the citric cycle, it actually gets used for gluconeogenesis in the cytosol.
Sites, so it pumps back the other direction. That's
Yeah,
cool.
it ends up getting used.
Oh, that's neat.
This is your presentation, you don't
Yeah.
Another part of this that I thought was interesting about our oxaloacetate, does that structure look familiar at all? Our oxaloacetate? Actually, it's not the oxaloacetate. Which one did I want?
Succinate.
Shucks. I was hoping there was a closer parallel within these. I'm not seeing a closer parallel. We can look at some amino acid synthesis coming out of these. We've got a lot of carboxylic acids, and if we drop in a transaminase, we can convert.
Transaminase?
Yeah.
Yeah, it's in the end.
You can convert the carbonyl into. You can convert the carbonyl into an amine, and you've got your amino acid popping out of that. Okay, so that's a part that I haven't looked at carefully enough yet to tell you which piece it comes from, but that's what I thought I saw in oxaloacetate, but apparently I did not get enough sleep last night because I don't see that. What happens in our next step, in our citrate to cis-aconitase? We formed a double bond, so is this a redox reaction? It's an elimination reaction, not a redox. We don't need an energy sucker. We don't need some energy intermediate to push this. What we're going to do is have probably some basic atom from your protein, pick up the hydrogen, and kick out, wouldn't probably be OH, probably pick up another hydrogen. My only answer to that is biochemistry. So officially, yes, in a real environment, we'd have to protonate it again to make it a better leaving group, but that doesn't mean that the protein isn't facilitating that leaving on its own. Okay. Yep. So that will get us our cis-aconitase, which gets us that double bond. So no redox. All we're using is the enzyme to facilitate an elimination reaction. What happened to the next step? Yep. So we did what type of a reaction? It's an addition reaction. What did we add?
Electrons.
Close. We actually added water. We added a hydrogen to one end of the double bond and an oxygen to the other. There you go. Exactly right. What we did was swapped where the oxygen was located. Okay. If we compare citrate to isocitrate, all we did was pick up that oxygen and move it from one carbon to the next. Okay. Why did we do that? We need to facilitate the proper alignment around those acid functional groups so that we can kick out CO2. So that alcohol that's floating back and forth is really facilitating the removal of our CO2. Depending on where it's located, it can actually allow for that process to pop off. Okay. What happens is we move from isocitrate to alpha-ketoglutarate. We got rid of CO2, so added hydrogen. There's a lot of stuff going on in this step.
You added another
Oh.
Yeah, we've got a carbonyl group coming out of that, which means we started with an alcohol and went to a carbonyl. That is,
I don't know.
Well, that hydrogen moved over.
that's an oxidation reaction. The hydrogen can't move over because if we look at eliminating our CO2, the electrons come down, what charge does our carbon pick up?
Oh.
Negative. We can't shuttle.
But doesn't that just grab the hydrogen that's
If we push the electrons down to form our carbonyl, the hydrogen's coming out as a negative, or as hydride. We can't just get a hydride shift because it's a negative negative. Okay? So there's a lot of stuff actually going on in this reaction. This is a redox step. Okay? We do have that hydride floating out there. So we will need NAD plus to act as an electron acceptor. The steps in this, I won't go into too much detail because I'm afraid Heather and Abby were potentially talking about this one, and I think this one's easier to understand than some of the other ones that require NAD. And you guys didn't respond to me, so I just kind of won't talk too much. Yeah, I emailed you. 2 different places. It's okay. You'll form, we'll just go through the basics of it. You'll form the carbonyl first. Okay. This reaction you might remember from OChem because what we've got here, that decarboxylation works because we form an enolate. Again, enolizable hydrogens, or it's not really an enolizable hydrogen in this case, it's actually the decarboxylation that is facilitated by an enolate composition. Okay. So you're right, I heard you saying the enolate, yeah. I know, no one was ready for it yet. I don't think people were ready for it yet. But what we are looking at is an enolate intermediate. That intermediate can go back and pick up hydrogens, and we end up with our alpha-ketoglutarate. Okay. What happens in the next step?
CO2 goes away.
We lose another CO2. We do have a sulfur coming, and where's, let's highlight it, where's this carbonyl? Purple is an awful color, let me try that again. We'll go orange. You guys see that orange one?
It's even worse. There you go. It's all right.
It's even worse. You guys just said it was bad enough that you guys could see it. Where's that carbonyl in the alpha-ketoglutarate?
Yeah, where do you see?
It's the same one that's circled.
It's the circled one. So if we go through and look at the loss of CO2, our CO2 is happening right here. Interesting. That carbon becomes negatively charged. What? We again have a problem with putting too much electron density on that atom. What's going to facilitate this reaction? It's almost an identical process to our pyruvate mechanism. Okay? Needs the thiamine, and the thiamine will attack the carbonyl first, okay, which is what's going to allow for our polarization and allow for all that electron transfer to shuttle around. We also have to bring in the lipoic acid. We'll also have to bring in the FAD, and we'll also bring in the NADH.
So the same thing.
Okay? It's almost identical as far as the mechanism goes. If you go back and take a look at our pyruvate structure, okay, and we converted, if we look at where the chemistry is occurring, we're looking right here versus right. What's the difference between pyruvate and acetyl-CoA versus what we just did? Nothing. It's an identical pathway with a different substrate. Because the substrate's different, what does that mean about the enzyme? It's also a different enzyme because it has to fold around the rest of that structure. Yeah, it's a bigger structure, and we also have another polar end to it, so we'll need something to actually kind of coordinate that and keep it back out of the way. Okay? We are definitely at time, but we will go back through all of those and finish out the rest of them whenever I get a chance to talk again. Yep. So what ends up happening is you end up shuttling that oxygen through the structure to facilitate the CO2 kicking out. Which I guess we should have mentioned back with the pyruvate, because we lost CO2 in that case too.
So we need to understand all of that.